organizers for inviting me. So to those of you who actually came for my alternate talk, which was called Interpretable Models of Cortex-Wide Calcium Imaging Data, this was actually what I was originally thinking of talking about for this talk, but I changed my mind. But for those of you who did come for this talk, I'll give you a one-slide overview of what this was going to be about. This was about how do we actually integrate neuroanatomical knowledge with large-scale neural activity, and that really, in my mind, was integrating prior knowledge with actual data. And this was really interpretable dimensionality reduction. And at the heart of this is that understanding that interpretability really comes from any prior knowledge you have about the experiment, about the data. And specifically, I was going to talk about methods to make sense of wide field calcium imaging data that involve using prior information in the form of an atlas, a region-based atlas, to get region-based self-localized components in the data, which make the decomposition both interpretable and robust. And that was also going to include some correlates to behavior after that and how this neural activity affects behavior. If you would like to hear about that, please come find me in the lunch break, and I'm happy to give you a one-on-one presentation. I already have the slides. However, I did change my mind and decided to talk about something unpublished and that I'm currently very excited about. I want to talk about how we make the same movement across different speeds. And this work is joint work with Abigail Russo, John Cunningham, and Mark Churchland. Okay, so let's get started. So I want you to consider the case where you're practicing a new dance move. So let's say, for example, the waltz. So you first practice it slowly, so you get the steps right. And then once you've learned it, you can flexibly speed up the movement and make it at the speed that you'd like. So how does the motor cortex do it? There are many other movements with this kind of where this kind of strategy applies. You learn slow and you make it fast once you've learned it. So I want to address the question of how the motor cortex might be achieving this goal. And the first thing that I want you to notice is that even though you might be performing the same movement at different speeds, your muscle activity necessarily changes while it changes fairly drastically when achieving these movements. So take the case of cycling slowly versus cycling fast. Not only are the muscles contracting faster, they may also have higher amplitudes and different patterns of contraction. However, once we have learned the movement at one speed, we want to be able to generalize to another speed. And thus there needs to be somewhere in the brain that can have a continuous representation of how the speed maps into these varied muscle activity patterns. Is that in the motor cortex, and how exactly does the motor cortex do it, if so? These are the questions we wanted to answer, and for that, for studying these questions, Abby Russo trained 2 monkeys to cycle through a virtual environment while tracking a target. The target was continuously moving at different speeds, and in addition, depending on the color of the landscape, the monkey cycles forwards or backwards to produce the movement. Now, you may be familiar with the cycling task from Abby's 2018 paper, but note that in that paper, the monkey was actually going at the same speed for maybe different cycles, but constant speed. And this is in fact a different data set, where one in which the monkey's speed changes continuously while tracking the target. At the same time, Abby also recorded single unit neural activity from the motor cortex and intramuscular EMG. So we first take a look at the muscle activity. So here I'm showing you the activity of one of the muscles, the deltoid, during forward cycling. Now this is a rhythmic movement, so we chop it up into cycles. And this happens to be the slowest movement, and the fastest movement looks like this. And as expected, the duration of the cycle is of course shorter since it's a faster movement, but more than that, the pattern of activity changes as well. And similarly, we have all the other speeds in the middle. We bin the activity into 8 different speeds. Okay, so here I'm showing the different two other muscles, but in total we have many more than that. First of all, we wanted to ask whether we can guess or predict what the motor cortex activity looks like during this muscle activity. So we wondered whether single units in the motor cortex might be encoding muscle activity or kinematics. And in fact, the data doesn't exactly show this. I'm here showing 3 example neurons, but there's no good relationship between these single neurons and the muscle activity, even in general. And the neurons don't seem to be doing velocity encoding either. However, so the single neurons do not in fact encode single muscles, and I'll go into that a little more deeply in a bit. However, even though this encoding hypothesis did not hold true in this data set, it provides a very good backdrop in that it gave us a clear quantitative prediction that we could make about the neural activity straight from the muscles. So we wanted to find such clear predictions using a dynamical systems framework. So for that, we move into state space first. So here I'm representing the muscle activity in the first 3 PCs. And we ask ourselves, in the dynamical systems framework, we ask ourselves, what would a network do? So here, recurrent neural network, but what would a network do? Or WWND, as I like to call it. So we used a recurrent neural network to simulate the activity. We can test that directly, right? With speed as an input. So the activity of the network's nodes seem to actually follow a very clear structure, which in fact looks very different from the muscle activity structure. And this is one example network, but in fact, I trained more than 50 models, and in fact, all of them follow a very similar structure. This is a very robust finding, and I'll go a little more deeply into that structure in a bit. But basically, there seem to be extra signals generated in the network that look very different from the muscle activity. And in fact, these signals are very important. And how do we know that? We can actually decode the neural activity, single neuron activity, with the network activity better than if we had just used EMG signals. So here I'm showing you an example neuron's firing rate in white, and the RNN PCs 1 through 3 are used to fit that firing rate curve, and I'm comparing that with the EMG PCs 1 through 3 fit. So here you can see very well that the example neuron's firing rate is fit much better using the RNN. And in general, this is not just true for one neuron, this is true for the entire population of neurons. So here I'm showing you the single neuron fits, and you can see very well the RNN fits that single neuron activity much better than if we had just used the EMG to do it. Okay, so that should convince you that the RNN model is a good one. So what does that mean? It can output EMG activity. It can fit single neuron activity. However, I could have done these 2 things with a very complicated input-output function as well. I could have done this with a 10-layer deep neural network that may not help us in understanding the computational mechanisms that are going on. So for the purpose of this conference, especially, that would not help us with interpretability. So here we want to get a sense of why does the model act the way it does, and what characteristics of the model can we actually expect to be present in the data. So I want to go into that, but also really talk about how geometry in high-dimensional spaces is becoming more and more important to figure out computational mechanisms. So, for example, Monte Cicillo's line attractor for choice generation, the Fusey Lab's positioning of context-dependent placement of high-dimensional firing rate activity, and very recently, as we heard in the main meeting, the Jazayeri Lab's curved neural trajectory to implement Bayesian computation. In all these cases, the geometry of the data and of the models in high-dimensional spaces turns out to be very important to figure out, to understand computational mechanisms. Okay, so what are the important geometric features in these RNN models that are helping us figure out the structure, which structures of this model are important? So to really get into that, what do we actually see in these models that are consistent across all the models that are fit? And in fact, we see very clearly 2 geometric features that are not present in the EMG data. And these geometric features are big circles in dominant dimensions. So here you just take the top 2 PCs. You see very clearly these big circles. And another geometric feature is the separation between these trajectories at different speeds. And why might that be happening in the model? So to really unpack that, I want to use a data-driven metric, trajectory tangling, in order to assess robustness to noise and really figure out why the model is acting the way it does. And why am I using this data-driven measure? In fact, we can actually measure that in the data, and that will help us figure out why the data might be acting the way it does as well. So what is trajectory tangling? It is, as it says, a data-driven measure to assess robustness to noise. What are we looking at? So take the system, the state of the system, x of t, at time t, and out of all the other time points, check which has the largest derivative, the largest difference in derivative, in fact, from x of t, but is the closest in state, here t prime. So basically, if it has a large difference in derivative, it better also have a large difference in state itself. Otherwise, a bit of noise can knock you over to another part of the trajectory, and you will have a very different evolution of the state. And so here, I'm showing an extremely tangled point, high trajectory tangling. This is the actual metric, the quantification of that intuition. And these are some trajectories with high trajectory tangling. And these, in fact, are some with low trajectory tangling. And I want you to note that, first of all, the rhythmic trajectory with the lowest tangling is a circle. And that brings me to, well, what does this have to do with the motor cortex and muscles? The hypothesis is that the motor cortex, in fact, has a very low trajectory tangling, since it is perhaps the one driving the muscles, and it is the one which is acting fairly autonomously, perhaps, in this kind of a task. Okay, so here I'm showing the model, again, the network activity, the actual activity of the nodes. And this gives us a concrete reasoning for the first geometric feature. So I told you that the trajectory with the lowest trajectory tangling is, in fact, a big circle. So here, this is exactly maybe the reason that we get this, the same big circle in high D space for these different speeds. Now, I do want you to note that if these were exactly the same big circle in high D state, state space, we would have a trajectory tangling issue because these would be going at different speeds. And remember, this the quantification was, again, the difference in derivative as compared to the difference in the state. So if these were all overlaid on top of each other, we would have that trajectory tangling problem, which brings me to the second geometric feature, separation between trajectories. And that, and this may help keep tangling low across speeds. Okay, so this is exactly the concrete predictions for the data. We have two concrete predictions, big circles, dominant dimensions, and a separation between trajectories. Now let's see in the data whether that holds true. And moreover, we will also be able to calculate the trajectory tangling in the data itself. So to check the first prediction, we take the monkey cycling forwards at different speeds. And as a comparison point, I'm first plotting the muscle trajectories here. So each plot is for the trajectory at one speed in its own PCs. And we see that the muscle activity is, in fact, changing patterns very drastically across different speeds, and also seems to have very high trajectory tangling at certain points in the trajectory. Now let's get to the neural data. So here, in fact, no matter the speed, we always see a big rotating circle in our data. So this is, in fact, very clearly showing that first prediction that I talked about of big circles in our data. And we also note that the fact that these are circles also keeps the trajectory tangling very low in the data itself. And here I'm overlaying all these trajectories for you to just show you the similarity between all of them for the neural data, as well as the differences between all of them in muscle data. You see really well that the pattern of the muscle activity changes across different speeds. And we can quantify this. I'm not going to take you through that quantification, the fact that these are big circles and very similar across speeds. But basically, this geometric feature, number one, has been, is seen very clearly in the data. And you also see the low tangling per speed. Now remember, if these were exactly the same trajectories overlaid with each other in high D space, we would have that problem of trajectory tangling. So we expect now a separation between trajectories. Let's see if that's actually true in the data. So in fact, the neural data in the top two PCs, now this is in the global subspace considering all the speed trajectories, in the top two PCs does seem to be fairly overlaid. However, as soon as we get to the third PC, we see very clearly a separation between the trajectories at different speeds in the data itself. And this is in fact a large separation, and we can really calculate that in high D space as well. This is for PCs 1 through 3, we see that, that separation clearly, but now in 10D as well, we can actually just calculate that distance between a reference trajectory and all the rest of the trajectories. And we see very nicely that for every theta, so for every phase in the cycle, we have the distance being monotonically increasing as a function of the speed, almost every theta. And now if these were completely straight lines, you see that they're not exactly straight lines. There's some curves at that harmonic. But if these were completely straight lines, there would be a pure translation effect going on. So all the trajectories would be purely translated from each other. However, they're not. So what else is going on? We can actually quantify the tilt in the data as well. So we see that the data is actually moderately tilted into different subspaces across speeds. To quantify that, we actually just quantify the variance explained of all the trajectories by projecting into the slowest PCs. And in fact, this drops fairly consistently, but monotonically. And so if this were a pure translation, this would just be at 1 the whole time. However, it's not, it's moderately tilting into different subspaces. We can do the same thing for the fastest PCs, and we see a very similar thing. They're not completely, the fast and the slow are not completely orthogonal to each other. In fact, they're nowhere close to that. So really, there seems to be, the picture I want you to have is that they're translating, but also tilting into different subspaces as a function of speed. Okay, so I showed you that the neural data is very well separated between speeds, but why is there a separation? And in fact, I'm now overlaying the actual trajectories now with their tangling values as well in the global subspace. And you can see that the tangling is very low for the neural data as compared to the muscle data. And that's exactly what we think is leading, the separation is leading to that low tangling. And in fact, we can directly test that. So here, I'm taking the within speed tangling, so that's actually just considering the tangling across just the speed, every speed of the trajectories. This is for the muscle data as compared to the neural data. You see that the muscle data goes very high as compared to the neural data. And now I'm quantifying the 99th percentile of just the neural tangling. And to get at this question of is it the separation, we first calculate almost a worst case. So that's basically taking a reference trajectory at one speed and just speeding it up, but really overlaying them in high D state space so that they're all just going at different speeds, but very close in state space. And that actually drives the tangling up by more than 2x. Now, what does the tangling in the global subspace look like? It's actually nowhere near that. It only rises a tiny bit for the neural data, whereas you can see that it rises quite a bit for the muscle data. And now to get at the question of is it the separation in the data that's leading to this low tangling, we can just artificially remove that separation and test that exact thing. And as you can see, the separation removed tangling does rise almost as much as the worst case. So it's really the separation in the data that is leading to that low tangling. Okay, so that kind of basically quantifies our second geometric feature. We see very clearly that the separation between the trajectories is a thing leading to low tangling across speeds. And that is kind of the last bit of that model where we can really understand the computational mechanisms of this RNN that's leading to sort of fitting our neural data really well. So this gives us a model that gives us all three things. It can output EMG activity, fit single neuron activity, and also we can actually understand the computational mechanisms underlying it. Okay, and with that, I want to acknowledge the people who made this possible, which is very heavily Abby Russo, as well as my mentor, John Cunningham, and collaborator, Mark Churchland. And thank you all.
Out of curiosity, what was the training method for the RNN?
Yeah, this was just backprop through time, vanilla RNN.
That was really neat.
Thank you.
I was just wondering, you mentioned at the beginning, well, I was wondering about the relationship between the neural activity and back to the either neuron activity or the RNN activity of the motor activity. And it seems that since the RNN fits the EMG activity, that there then is there a linear mapping then between the trajectories of the RNN and
That's right. Yeah, I just use a linear decoder from the RNN to the neural activity, as well as a linear decoder from the EMG to the neural activity, to get that comparison.
So they can then be done with the neural activity and see direct relationships between the neural activity. Keep this in mind, or
So like a vice versa, basically use neural activity to fit the model as well as the EMG. No, maybe I'm getting this wrong.
It seems that the neural activity now resembles, in some sense, the RNN fits the motor activity.
That's
So
right.
that would be the mystery between. Is it then possible to make that connection there?
Oh yeah, so the neural activity does decode the EMG really well.
Oh, it does.
Yeah, but the model does as well. Yes.
I see that, right?
Yeah, that's right. But the other way around is not true. So the EMG is not very good at decoding the neural activity, and it's not very good at decoding the model either. That's right. Yeah. Thank you.
I was wondering if you could it be possible that neural activity is encoding something like the kinematics? It seems like it's a very cyclic task.
Yeah, so in a previous study, people had really studied that, but basically, if the neural activity was decoding directly the position, for example, at time t, you would also see that work very well for forwards as well as backwards. And actually in the neural activity, we see very clearly that forwards versus backwards are almost orthogonal to each other. So it's really not basically it's not only decoding the position at time t or the kinematics very clearly.
It could be like
It is very cyclic, that is true, but this is another explanation for why.
What about velocity? Have you checked that?
Yeah, so again, the same thing applies. The orthogonality means that it's not good at decoding both forwards and backwards.
I wonder if the neural activities are encoding velocity. I wonder if you were able to decode to a better job without even using RNN, but just using the EMG and the different time derivative of EMG to reconstruct the neural activities.
The time difference of the EMG.
So time derivative.
Yeah, we didn't check exactly that. We didn't check the EMG and the time difference and all these other things. The velocity, however, again, as I said, the neural activity between forwards and backwards is almost orthogonal to each other. And that means that any decode from a position or a velocity signal will not be very good, and that's explored a little bit more in Abby's 2018 paper, so I didn't go into that too much. Yeah, thank you.
If you look at single trial activities, do you see any relation with the manifolds that you're looking at?
Yeah, this is all trial averaged activity. We haven't explored the single trial activity too much. These were recordings done separately, so it's difficult to do that. But yeah, maybe in a follow-up study that would be a cool idea. Yeah.
I have like a meta question given the workshop. I really like how you kind of extract computational principles from the RNN and relate them to neural data. Do you think there's any value in like trying to interpret how the RNN is generating these things? Like, is there value in looking inside of the RNN?
Yeah. No, I think there is, and I'm currently working on that kind of a project. But so for the purposes of this talk, for example, we really wanted to get at what is happening in the data as well, so come up with metrics that are actually testable in data, including trajectory tangling, and the geometry as well. So that's why we resorted to this. But yeah, it'll be pretty interesting to see exactly what in the model is leading to this structure. And there are other methods for that, like the fixed point finding tools, etc. Yeah. Yeah, it's great.
Okay, great.
Thank you.
Thanks, next speakers are